First of all, I hope the audience still has some caffeine in the system in order to be alert. And today I'm here to talk about decoding coffee cardiometabolic potential and try to also show some structure-function relationships that we have obtained with our work. Of course, this was not possible if the organization would not invite me to present this work, okay? So I would like to acknowledge in all the presence, and I think all the talks are very complementary, which is quite nice. So I'm talking about cholesterol because I'm very interested in cardiovascular diseases. Why? Because they let people die, and because the degree of incapacity that people can suffer with these diseases is really a social problem. So if we find strategies that can solve this problem, is very interesting. And there are huge losses. Mostly due to high levels of cholesterol in blood. Elevated LDL cholesterol is the reason why 3.8 million deaths each year occur. So this is really a problem. And we are not talking about the incapacity that these people can have. Okay, so to understand a little bit about cholesterol, I bring this scheme. I hope it's not too complicated, but I would like to understand that cholesterol is mostly produced at liver, or a big part, 2/3, and 1/3 is coming from the diet, and it needs to be solubilized in the intestinal lumen in order to be absorbed, okay? And over here, there are some functional food that can act in order to decrease this absorption. I'm not talking about pharmaceutical that exists and everyone takes, or the people that has problems like statins or cationic resins, or ezetimibe, so inhibitors of cholesterol transporters. I'm talking about food that in the beginning could be important to reduce these levels and to control these levels at the beginning. And so we try to avoid this absorption in order to have less cholesterol in the bloodstream and supposedly less cholesterol in the system, so less potential to have arteriosclerosis. The other thing also that is important is the role of bacteria fermentation, namely on fibers, which can alter and produce short-chain fatty acids, but could also affect the production of bile salts, which we call secondary bile salts, which are more hydrophobic, and so they can solubilize more cholesterol and could have an impact on cholesterol metabolism. Okay, so I'm looking to bioaccessibility, bioavailability, and fermentation. So nowadays we have this functional food, but they have some problems. They are not taken after the major meals of the day, where probably they were more active. They are mostly based on hydrophobic nature, so everyone knows the sterols that we take in yogurts or beta-glucans that we take in cookies. And there is a need for encountering new strategies to solve this problem or to help on this problem, and also a new opportunity for industries. What about coffee? Why not coffee? Well, coffee is taken after major meals of the day. We can somehow modulate the content of these compounds. It's also an hydrophilic matrix, can be taken after the major meals and could be a quite interesting matrix, okay? So, okay, but there are some problems. We have caffeine, we have diterpenes, but as we have already seen, we can change this and we can modulate these compounds, and so, but we have polysaccharides and melanoidins, which could be quite interesting on this subject. So basically, what we try to do is to fractionate and chemical characterize some coffee high molecular weight compounds from coffee, modulating, roasting, and also grinding, and also evaluation the cardioprotective potential of this modulated coffee and these high molecular weight extracts. And even if possible, we try to find structure-function relationships. Okay, so this is more or less the methodology. We do the coffee processing, we characterize, we extract, fractionate, we do some cholesterol bioaccessibility assays. We also do the permeation through CoA2 cells, mimetizing the intestinal epithelium. We try also to see in fermentation using human fecal microbiota, and also we try to assay using mouse models. We would like to go to humans one of these days. Okay, so But does coffee has this potential or not? So what we did was, okay, we use a very basic system where we have a bile salt and cholesterol level, and we try to address, does this really reduce cholesterol? And what we see is that using available coffee, that we had a reduction in the solubility of cholesterol, which was interesting. But why we have this reduction in solubility? When I'm talking about solubility, you may understand is when it's bioaccessible or not in the intestinal lumen to be absorbed, okay? So, and what we notice here in the right is that with a control, we have a certain amount of cholesterol. In presence of different extracts, we have a decrease in this solubility. We also try black tea and Luisa Citrodora tea, but we didn't see that. So it was not, it's something really from the coffee. So we tried to understand what was the reason behind that. And the reason behind that was that our extracts that were rich in polysaccharides were sequestering the bile salts. There was a relationship between the sequestration of bile salts similar to what happens with a cationic resin that is already a pharmacological molecule that you have that can sequestrate the bile salts because they are negatively charged. The resin is positively charged, and so there is this sequestration. And so our fibers from coffee were doing similar stuff, not because they are cationic, but because they have hydrophobic connections. Okay? So, and this was the relationship I was talking about. Okay, so sum up, commercial coffee show hypocalcemic potential, cholesterol bioavailability is affected by the presence of these polysaccharides, fibers, you may say, and the mechanism would be the sequestration of bile salts. Nice story, but can we modulate this sequestration? So the next step was, okay, let's go and extract, fractionate, roast different roastings and collect these fibers. So this is the main procedure. We all do the characterization. I will not go into detail in this characterization, but basically we could do by glycosidic analysis, we can see the difference, the structural differences on these fibers and try to see if these structural differences could be related with the bioavailability we were saying. Okay, so over here we have another assay with different roasting and different fractions, so melanoidins, Arabinogalactans, galactomannans, all from different roastings, so light roasting, medium roasting. We went to a coffee company in Portugal, a very small one. Can we use your facilities? And they say, yes, you can. So we start to doing this kind of experiments. And basically, we got some interesting results. The degree of branching could be related with the cholesterol that we were seeing. So basically, if we have an increase in degree of branching, we could affect cholesterol bioavailability, which was quite interesting at this point. So I think I already said the main conclusions. I can skip this one. Okay, so what about bioavailability? Until now we are talking only bioavailability, the solubilization of cholesterol. But does it passes through CoA2 cells? So we try to do this in model cells, mimetizing intestinal epithelium, and we try to see how does this behave. So over here we didn't use the same bile salt because the one previous was cytotoxic. So we need to take care of this. So this picture here shows that in the presence of coffee and cholesterol analog, in this case we use dihydroergosterol, that is a fluorescent, so it was able to see the passage through the CoA2 cells. I'm seeing some faces a little bit. If I'm too scientific, please tell me. What we see is that we have this passage through the bilayer. So in presence of the coffee, over here we see the solubility, so we have different roasting levels. So light roast, dark roast, okay, with different grinding. So we were trying to see if this roasting and this grinding could affect the potential of this coffee. And it really, we see that. And then we try also to see what would be happening. And what we see is that there is this precipitation of cholesterol, because if cholesterol is not solubilized by the micelles, it tends to precipitate and it goes be excreted. And we see that, the deposition of these crystals, in this case of the sterol analog. And then we also see over in the right that there was a decrease in the bioavailability. So you have the apical, you have the cell lines, and then the basolateral. So if you have the decrease of entrance in the basolateral, you are seeing that the bioavailability is not occurring, which is good in the presence of these fibers. Okay, so in this case, dark coffee. But, and what about the fermentation? Can fermentation affect or can have potential to improve the cholesterol? So here what we study was, this is a co-work with Professor Castillo. We use melanoidins and arabinogalactans that we extract from coffee, a special kind of coffee, and then we see the metabolization of these compounds. These fibers, they do not suffer any alteration during the gastrointestinal, but once in the colon they change. And what we see is that arabinogalactans were much more metabolized than, for instance, melanoidins, okay? And also that the production of secondary bile salts, which are known to solubilize much more cholesterol, and it's a problem because if you solubilize more cholesterol, they can be going through heteropathic recirculation, so they are going back to the liver, coming from the blood to the liver. If you have more of these bile salts, you are solubilizing more cholesterol, so more cholesterol can be bioaccessible. So the idea is that we need to reduce the quantity of these bile salts. They can also be promoting other diseases, okay? So this was a very interesting also result. Okay, the last thing that I would like to tell you is that we also try to do some experiments in animal models. So we use this animal model that you have here during 7 weeks. This is with a partner from Lund University. And what we see is that the presence of coffee, there is no weight gain. Not in the case of melanoidins, that doesn't happen. And the interesting part is that we saw that there was an increase in cholesterol, but mostly the cholesterol that was increasing is the HDL, so which is good for our health, heart health. Okay, so coffee and melanoidins extract affect body weight of mouses in controlled diet and high diet, and coffee melanoidin extracts show an accretion on HDL profile in a high-fat diet, because we use normal diet and high-fat diet. Okay, as a main conclusions, what we can see that commercial coffee and its extracts have potential to affect bioaccessibility, okay, and we establish some relationship. We can have sequestration of bile salts, and this is important relating to this metabolism. Cholesterol bioaccessibility can modulate if changing in roasting, in grinding, the extraction. So we are looking the compounds that are present in the beverage. We can somehow modulate them in order to improve this potential. Coffee affects the bioavailability of these compounds using these CoA2 cells, and also fermentation has also this possibility. So we need to look this in a holistic way. So we cannot only look to bioaccessibility, bioavailability, you need to look also to fermentation for other issues, as we already have heard today. And also coffee and melanoidins affects HDL profile of mouse fed with high-fat diet, showing cardioprotective potential. Nothing of this would be possible if we don't have collaborators, if we don't have PhD students that are working hard on this, and also if we don't have funding. I would like to finish by showing that I have a dream, okay? My dream is to have a fight sterol company which work with coffee, would be the first product. I'm waiting for some people from the coffee industry that would be interested on this project, and we could dream together, because this is all about dreams. And I have already the slogan, fight sterol coffee, what else? Very original, as you know, but I have this dream. Thank you. Thank you very much.